In today's class and video, we are going to look at an important enzyme that we are going to use in our mini project, acid phosphatase. Basically, we are going to look at its isolation and its characterization. So we'll start our introduction by looking at what the enzyme phosphatases are. And first of all, what I want you to know is that our enzyme acid phosphatase belongs to this group of enzymes. So phosphatases are enzymes that catalyze the hydrolysis of phosphate monoester bonds with the release of inorganic phosphate. So this is just a general reaction showing how the enzyme phosphatase catalyzes the hydrolysis of the phosphate monoester bond of a substrate to produce an inorganic phosphate as seen over here. The second reaction over here happens to be a specific reaction where a specific type of enzyme is used, and this is alkaline phosphatase, and also a specific type of artificial substrate which is p-nitrophenol phosphate is used. Now, so over here we can see that the enzyme alkaline phosphatase is used to catalyze or hydrolyze this artificial substrate p-nitrophenol phosphate to p-nitrophenol and an inorganic phosphate, as also seen over here. Now, so what you also have to note is that phosphatases are widely distributed in nature. Some of them are highly substrate specific, and in such cases it is usually not difficult to assign a physiological function to them. So example is fructose diphosphatase, which is used to catalyze the intermediate reaction in gluconeogenesis. Another group of phosphatases is also found in cells of most organisms, and these enzymes have extremely broad substrate specificity and are generally classified as acid phosphatase or alkaline phosphatase based on their pH optimal. Now let us look at acid phosphatase. So what we have to note is that acid phosphatase is an enzyme that catalyzes the hydrolysis of phosphate esters in an acidic environment. The key note is in acidic environment. So acid phosphatase exists in several biological kingdoms, including plants, animals, fungi, and bacteria. And acid phosphatases are a family of enzymes that belong to the hydrolase class, and they are specifically grouped together because of the shared ability to catalyze the hydrolysis of orthophosphate monoesters under acidic condition. They are also widely spread in nature and can be found in many animals and plant species. So examples: the seed of plants are particularly rich in acid phosphatase, and also the amount of phosphatase activity increases sharply upon germination and decreases as the seedling develops, because plants use this enzyme to obtain their energy from phosphate by hydrolyzing phosphate esters during energy metabolism. So now let's look at the isolation of acid phosphatase from maize germ. So we want to isolate 100 mils of the acid phosphatase from a maize germ. So this is the protocol that we are going to. So we first have to weigh 40 grams of the germinated seedling and blend with 50 mils of ice cold water. We transfer into a 100 ml beaker or bowl on ice cube. We wash and blend with extra. We wash the blender with extra 50 mils of the ice cold water and add to the 100 ml beaker or bowl. We allow to stand with frequent stirring for 30 minutes on ice cube. So after that, we centrifuge at 10,000 gravity for 20 minutes at 0 degrees Celsius. Then we decant or filter through Whitman #1 filter paper. So after doing this, we add 6 ml of Triton S100 as a detergent and stir gently to lyse the cell. So after the cell is being lysed, we pour the supernatant, which contains our acid phosphatase, into a clean vacuum tube and store in -20 degrees freezer. And what we also have to note is that so once a frozen fraction of the stored enzyme has been thawed out, it must not be refrozen. So now let us look at what the Triton S100 is. So Triton S100 is a commonly used detergent in laboratories, which is widely used to lyse cells, to extract proteins or organelles, or to permeabilize the membranes of living cells. So let's now see how Triton S distracts cell membranes. Triton S100 distracts cell membranes by inserting detergent monomers into the lipid membrane to permeabilize the membrane. And also Triton S can also distract cell membrane by 1. Distracting the lipid-lipid interactions, and 2. Disrupting the lipid-protein interactions and causing cell membrane rupture, and 4. Causing swelling of the chloroplast. So these are 4 ways by which the Triton S can distract cell membranes. Dilution of enzymes. So first of all, what I want us to note is that enzymes can be diluted with water in the laboratory. So now let us look at why water can be used to dilute enzymes when working with them. One is that water is not an enzyme inhibitor. 2, water does not change the enzyme pH. And 3, water does not affect enzyme temperature. And 5, the rate of enzyme reaction remains the same in water. So these are a few of the reasons why we can use water to dilute enzymes in the lab. So now let us look at the characterization of enzymes. So the characterization of enzyme involves the determination of its optimum pH, optimum temperature, the Michaelis constant, and the V maximum, which is the maximum rate at which enzyme catalyzes reaction. So these are some of the ways that we can study or characterize enzyme in the laboratory. And also, for their purification, the following methods can be used. So we have the ammonium sulfate fractionation, the ion exchange chromatography, the gel electrophoresis, and dialysis. So these are 4 ways by which we can also purify our enzyme in the laboratory. Thank you.